Okay, so let's move on. We can go directly to our next speaker for today, the second presentation for today, which is from Arrate Munoz Barrutia. Arrate is a full professor in bioengineering at University of Carlos of Madrid, known for pioneering contributions to biomedical imaging technologies and advanced image analysis, including notable work in 3D histological reconstruction and AI integration in bioimaging. She leads research initiatives in computational bioimaging, actively contributing to open source projects like Deep Image J and the Bioimage Model Zoo. She has received several prestigious awards and holds influential roles in the scientific community, contributing to both national and international research collaborations. In this talk, Arrate will introduce us to the importance of FAIR principles in preparing bioimage data and AI models for advanced research applications, focusing on creating FAIR datasets. We will explore the Bioimage Model Zoo as a practical example of how FAIR AI models enhance bioimage analysis accessibility for diverse users, from academic researchers to biotech and healthcare providers. Through this session, participants will see how FAIR practices in data and AI models can drive innovation and reproducibility in life sciences. So then I will stop share my screen, and Arrate, I think if you are ready, you can share your screen with us. Welcome to EU-OPENSCREEN Training School. We are very happy to have you here.
Okay, I gonna.
Yes, we can see your screen now, and now if you put it in full mode. Yes, yeah. That's great. Thank you, Arrate.
Thanks. So, hello, good morning to everyone. I would like to thank for the invitation and have AI for Life here in the school. And today I will present you FAIR bioimage data and artificial intelligence models, how to advance accessibility, innovation, and reproducibility in life sciences. Imagine computational biology as a landscape of innovation, but with bridges still under construction. On one side, we have the groundbreaking artificial intelligence models and methodologies. On the others, researchers and practitioners that are eager to use them. Our mission is to connect these sites, ensuring that bioimage data and artificial intelligence models are not only cutting edge, but are also accessible and reproducible. And AI for Life is actively building this bridge by providing tools, platforms, and collaborative opportunities that democratize access to these bioimaging resources. One of the critical challenges in the adoption of artificial intelligence and deep learning methods in life sciences is accessibility. And while artificial intelligence has the potential to revolutionize image analysis, there's still significant barriers. First, only a small subset of experts train and design artificial intelligence networks, limiting widespread adoption. Second, while some models are published in papers, so they are available as code or tools, but their usability varies widely. And this fragmented accessibility creates pressing questions. How can we enable tools to seamlessly exchange models? The solutions from AI for Life are to the development of initiatives. The first one is the Bioimage Model Zoo that is a repository of pre-trained artificial intelligence models. They are all stored in a standard way and allow the models to be shared, tested, and implemented across different tools and platforms. We also focus on creating artificial intelligence-ready data sets that follow FAIR principles and that are stored in the bioimage archive of EMBL, EDI. And finally, we are held open calls and challenges to encourage the community to collaboratively solve complex image analysis problems, fostering innovation and creating tools that are robust and scalable. One of the issues is that many imaging scientists, they do not code, and they rely on intuitive tools rather than programming expertise, which creates a gap between the availability of advanced artificial intelligence methods and the usability in everyday research. Second, even for those that has coding skill, most scientists don't fully understand deep learning, the technical complexities of neural networks, how to train them, fine-tune them, and deploy them can be daunting, especially for not experts. Finally, annotation, that's an essential step for training artificial intelligence, is already labor-intensive and difficult. This further limits the scalability and adoption of deep learning technique. So, how can we put the networks into the hands of the imaging scientists in a way that is intuitive, efficient, and impactful? So for us, the solution lies in creating tools and infrastructures that abstract the complexity of deep learning. And we focus on user-friendly platforms, well-documented workflows, and accessible model repositories like the Bioimage Model Suite to empower scientists to use artificial intelligence without needing to be experts in the field. And with these efforts, we aim to bridge the gap, ensuring that the potential of artificial intelligence in bioimage analysis is fully realized across disciplines. What's the Bioimage Model Suite? The Bioimage Model Suite is a community-driven open source repository of artificial intelligence models. The models are documented, cross-compatible, and ready to integrate into diverse workflows. Here you have the web page of how the web page of the Bioimage Model Suite look like. And the Bioimage Model Suite represents the practical realization of the FAIR principles. They are findable, accessible, interoperable, and reusable within the context of bioimage analysis. As I said already, the platform provides a centralized repository where researchers can explore, contribute, and utilize a variety of pre-trained artificial intelligence models that are specifically designed for bioimage analysis. It brings together the efforts from the leading labs and institutions, including Eurobioimaging, Enriquez Lab, Hub Lab, Crescu Lab, Obian Lab, and others, fostering a collaborative and community-driven approach. The first principles in action, as represented in the Bioimage Model Suite, are the models are findable, all models are indexed and searchable, making it easy for users to locate the tools they need. They are accessible, models are available with clear documentation and metadata, ensuring anyone can download and use them. They are interoperable by adhering to standards. The models can seamlessly integrate with diverse software platforms and workflows. They are reusable. Detailed metadata and licensing ensure that the models can be adapted, modified, and applied across a range of research scenarios. In this way, the users can explore the models, test them directly, and contribute back to the repository, creating a dynamic and either evolving resource for the bioimaging community. Oops. Okay. The first step is the model creation and upload. The journey begins with the creation of the artificial intelligence models for bioimage analysis. Researchers, they train their models, link them to the data sets, and document their processes through Jupyter notebooks. These resources, along with the detailed descriptions, are then uploaded to the bioimage.io for sharing with the community. The bioimage.io serves as a central hub where users can explore a curated collection of the models. Each model is accompanied by a comprehensive model card that provides information as the training data, the documentation, the associated data sets, and if there's a related publication. Models are validated and are easy to download. They have clear licensing and, as I said, the DOI assignments for proper attribution. Once downloaded, the models can be integrated into various user-friendly software tools, the community partners of the Bioimage Model Zoo, and here we have a number of them. They are growing on time, like Elastic, DeepImageJ, Therocos, Stardisk, QPAT, Enjoy, etc. The platform supports key applications, including the segmentation, the classification, artificial labeling, denoising, and super-resolution imaging. For advanced users, developers tools like Jupyter Notebooks allow customization and further refinement of these models, enhancing their utility for specialized tasks. This workflow enables researchers of all backgrounds, from basic scientists to computational experts, to access, use, and contribute to state-of-the-art models, fostering this collaborative and innovative community. Key features that make the model, the Bioimage Model Zoo, a standard platform are: it's community-driven. The zoo is built and maintained by the Bioimage community. It's open source and transparent. Foster collaboration about contributors from different fields and institutions. The researchers are allowed to contribute their models, ensuring a diverse and rich repository that reflects the latest advancements in the field. The models that are hosted in the zoo are designed to work seamlessly with various consumer software platforms, so their accessibility is expanded. Here in the cards, you can see the icons are the community partner software in which the model can run. And every model comes with comprehensive documentation, making it easier for users to understand, test, and deploy the models and their workflow. And they have the proper licensing, and the users have the selectivity to access their preferred consumer software and download accordingly. Finally, the platform facilitates easy sharing and application of pre-trained neural networks, lowering the barrier for entry and accelerating the adoption in artificial intelligence bioimaging. The Bioimage Model Zoo community partners represent a collaborative ecosystem of the tools and platforms designed to advance bioimage analysis by integrating a state-of-the-art artificial intelligence model into the research workflow. As I told you, the community partners include popular software like Fiji, IC, and QPAT. We are widely used for image analysis and visualization. We also collaborate with cutting-edge artificial intelligence platforms such as DeepImageJ, Stardisk, and Deep Learning for Microscopy, which make implementing deep learning model more accessible for the mind scientists. And also, we have initiatives. The partners are also Initiatives like the Human Protein Atlas, which provides data sets for developing and testing the models. Okay. What does the bioimage model zoo ecosystem include? The ecosystem provide access to the pre-trained model and the detailed model cars. Model car contains a trained model ready to be used or fine-tuned, example, input and output images, allowing users to quickly understand the model functionality, a cover image for easy identification in the GUI, and a detailed resource description file that specifies all necessary metadata, including architecture, input-output formats, pre-processing steps, and performance metrics. It also includes validator scripts, which help ensure that all the uploaded models meet high-quality standards and can be reliably used across various platforms. And the structure facilitates reproducibility and also the contribution of models by users. We have bioimage.io GitHub repository that holds the code, the specification, and the tools that power the platform. And in this way, we are providing with all the tools that we provide, we ensure that researchers can easily integrate the artificial intelligence into their workflows without needing extensive technical expertise. Okay. And the foundation of the bioimage.io platform is the ability to consume the pre-trained models. For those with more specific needs, the platform supports fine-tuning, and users can adapt models to their unique data sets or research requirements. Community partners like studies and tools develop in like Zero Cost or Deep Learning Everywhere facilitate this process, empowering users to customize models for enhanced performance. Once refined, the models can be re-exported and shared back with the community through the Bioimage Model Zoo, and this ensures this continuous cycle of innovation where improved models are accessible to others and that foster collaboration and progress. The model cars, you can see here examples. They are an essential feature of the bioimage.io platform, as they provide the information that the users need to select and use the model for their bioimage tasks. The model cards are important because they simplify the decision-making process for researchers by presenting all the relevant information in a single, easy-to-navigate format. And this transparency helps to assure that the models are applied effectively and responsibly. Here we have examples of model cards for tasks like nuclei segmentation, mitochondrial segmentation, and bacterial segmentation. And each has visual outputs and also detailed metadata that helps the user understand the expected results and the appropriate applications. One of the nice features of the Bioimage Model Zoo is the ability to test the models directly in the browser, making helping the non-coded users to try and test the models easily. Okay. So here we have the tested interface of the nuclei segmentation boundary model. And with just a few clicks, the user can load images, run the model, and compare the output to reference results. And this simplified, is a game changer for making artificial intelligence accessible. Here is a small video that represents the test. Let's see if it works. So you can load the image, run the model, and check what will be the output, either in test images or in your own images. Okay, now I gonna briefly describe BioPy, that is library and application also for accessible deep learning for bioimages. This slide provides an overview of BioPy. The key components are the input. BioPy accommodates diverse input types, including multi-channel images, 2D and 3D data. And this flexibility ensures that it can handle a wide range of bioimaging tasks, from basic microscopy data to complex volumetric data sets. The library supports various analysis tasks such as segmentation, classification, and tracking, making it a powerful tool for tackling bioimaging challenges across different domains. The BioPy integrates seamlessly with frameworks like the bioimage.io, Torvision, and custom pipelines. Its modular approach allows users to implement workflows that include pre-processing, post-processing, and everything in between. The front end. On the user interface side, BioPy provides options for both novice and expert users. Whether you prefer graphical tools or combined line interface, the library ensures accessibility for all the experienced devs. The library is a collaboration of several groups, and the main leader of the library, of the development of the library, is Ignacio Aranda Carrera, that works in the Basque Country in Spain, and Daniel Franco, that did his PhD co-supervised by Ignacio and myself, and now he has started working in Carmona's lab in Cambridge. Here, I would like to illustrate the flexible front-end options provided by BioPy. That there's several options for the users to access and to use the library. So from the non-expert users, the BioPy use a graphical user interface that makes it straightforward for researchers to interact with the library without requiring any programming language. We have also the option to use Google Colab to run pre-configured workflows directly in the web browser with a minimal setup. For advanced users, we have Jupyter notebooks and also Docker containers to bring portability and scalability to the table. And finally, for the most advanced users, they can use the terminal for full control and to work directly with scripts and tailored processes to their specific needs. The core strength of BioPy is the streamlined workflow or pipeline, which integrates pre-processing, deep learning models, and also post-processing. The pipeline begins with the input data. which can include images like microscopic data and associated level of CSV files, and the flexibility supports a wide range of bioimage analysis tasks. The pre-processing prepares the data for the deep learning model. Includes tasks such as the generation of 2D and 3D patches, normalizing intensity, representing problems, and applying filtering techniques. The core of the workflow is the deep learning model, which processes the data to extract meaningful patterns and insight tailored to the analysis task. And finally, we have the post-processing that refines the result. It can involve filtering, mosaicing, agglomeration, applying maximal separation, and calculating evaluation metrics to validate the output. And the output can be in the form of processing images or structured data that, like CSV files, making it versatile and done for downstream analysis. For users, the workflow is intuitive and straightforward, enable them to combine pre-processing, the modeling, and the post-processing without extensive technical experience. For developers, BioPy provides the freedom to contribute or customize any module, ensuring flexibility and scalability for the advanced applications. Here, we'll give a detailed view of the architecture and the processes powered by BioPy, showcasing how it integrates user-friendly front-ends with efficient backend for bioimage analysis. On the front-end side, we have this idea I gave before of the accommodation of user across the spectrum of expertise from GUIs to the terminal. And the input formats, we have a variety of input formats, including labeled datasets, CSV files, and image formats like NumPy, GIF, HDF5, and SAR. The compatibility ensures flexibility for the researchers working with different types of bioimage data. In the backend, we have a robust workflow that comprises this pre-processing, training, post-processing, and the pipeline is designed to be memory efficient and support multi-GPU processing, what makes it scalable for large data sets and complex tasks. And the pipeline generates the outputs that can be site-solved images, labels, CSV, and they additionally provide detailed metrics like area, location, circularity, volume, surface area, specificity, and these outputs enable quantitative analysis and facilitate deeper insights into the bioimage data sets. To configure the workflow in BioPy, we need this YAML file, which its task or problem just require a YAML template. The approach ensures that the workflows are standardized, reusable, and easy to set up. The YAML files enable easy prototype, just because instead of writing complex code, user can edit the text file to define the workflows. And that lowers the barrier for non-programmers while maintaining the flexibility for the advanced user to customize the pipelines. It's true that the YAML files come with a long list of keywords, but the users don't need to memorize or know them. But there's templates and examples provided in BioPy, web page and documentation to simplify the process and enable the users to focus on the research rather than the technical details. The YAML integrates seamlessly with the tools like Google Colab or the Jupyter notebooks, allowing users to launch workflow with the minimal effort. Here, I'm not gonna go into the video because it's like 5 minutes long, but there's a tutorial that explains how to import and export models from the bioimage model SU into BioPad. Now, I'm gonna introduce you to the bioimage model, SU, the bioimage.io chatbot. That is a community-driven artificial intelligence assistant that we designed to support integrative computational bioimaging. The chatbot provides real-time assistance, enable the researchers to interact with the bioimage.io and the resources seamlessly. It simplifies complex workflow and answers technical questions to make bioimage analysis more accessible for everyone. One of the biggest challenges in the bioimage analysis is the steep learning curve associated with the tools and with the workflows. And the chatbot aims to bridge the gap by acting as an accessible guide, enable more that is available anytime the researchers need help. And the project is a collaborative effort that involves partners in Ericsson, KTH, and the University Carroster in Madrid. And the chatbot is available in the link in the bioimage.io chat. And it provides guidance and troubleshooting tailored to the level of expertise of the user. The idea or the fact is that the large language models are transforming how we approach computational tasks and their potential to support bioimaging. And the large language model are versatile and can process various types of input data, including text, images, speech, structured data, code snippets, knowledge nuggets, etc. For the tasks that involve documentation, the large language model excel at summarizing, language translation, sentiment analysis, and classification. And also, they can assist in code generation, enabling researchers to develop scripts for processing and analyzing bioimage data more efficiently. For example, the large language model could summarize microscopy experiment results, classify types of bioimage data, or assist in drafting reproducible protocols. So, by integrating the large language models, we can reduce the cognitive load of researchers and allow them to focus on innovation and discovery. The challenges in using large language models, as you know, are the limited reproducibility with inconsistent answers to the same question, limited cutoff because in many occasions they are not continuously updated, bias that it depends on the training data, and the generation of hallucinations. So we developed the chatbot using retrieved augmented generation to provide highly relevant and accurate outputs and improve or get rid of the limitations that I have just explained. This slide demonstrates how the retrieved augmented generation can be used to enhance these large language models. by incorporating the community-driven knowledge and ensuring more accurate and context-aware responses. How the RAC works in this framework. First, we have a process of documentation, retrieval, and ingestion, and to ensure that the model has access to the most current and accurate information. The second step is that the retrieved documents are converted into embeddings using this embedding model like OpenAI's, and this embedding encode the context and the semantic of the documentation, making it easier to match queries with the more relevant information. When the user submits a query, it's also embedded and matched against the stored document embeddings using a vector database, such as files. And that ensures that the query is paired with the most contextual relevant documentation. The matched documentation along with the user query is provided to the large language model as additional context. This reduces hallucinations and improves the relevance and reliability of the generated response. And the response is part of a real loop that allows for interactive refinement, ensuring that the model continues to provide accurate and relevant answers over the time. In this way, we have a chatbot or an assistant that provides highly specific, reliable, and context-aware guidance in the bioimage workflow. The slide illustrates how this chat assistant is designed to integrate bioimage analysis documentation at books, also database and tools, creating a simple and intelligent user experience for the bioimaging community. We have designed the chatbot with 3 specialized roles. The first one is the bioimage seeker that is called Melban, and the module focuses on retrieving relevant documentation and data from the knowledge base. It is designed to effectively locate the most contextual appropriate information for the user queries. The second is the bioimage tutor that we call Bridget, and this component acts as an educator, guiding users through workflow, tutorials, and resources. Whether it's learning about a new tools or understanding a data set, the tutor ensures users are equipped with the right knowledge. And the third one is the bioimage analysis, Nina, that the module provides advanced analytical support, helping users design workflow, troubleshoot issues, or execute specific bioimaging tools with the artificial intelligence support. The assistant uses the React loop to iteratively refine the responses based on the user feedback and the context. It also considers the user profile, the conversation history, and the specific query to deliver personalized and accurate guidance. Now, I will show you how the bioimage chat assistant works. Here we have an example of the use of the bioimage seeker of Melbourne. That, for example, is able to suggest strategies like using informative tasks, clear model name, and representative images to improve the model visibility. That simplifies what will otherwise be a time-consuming and complex process. We have another example here of how we can use Melbourne to explore specific protein data in the Human Protein Atlas database. Here, for example, if you are looking for a database like cell images from GA phase, the assistant is querying the bioimage archive to provide you with the detailed information about the data set, the title, the accession, the description, and always give a link to the data set in the bioimage archive. The tutor, for example, can give you information about technical tools or aspects, like, for example, how the convolution works. And the bioimage analyst, Nina, is prepared to help you in the analysis of your data, and just by uploading data is running cell-post model to do the segmentation of the cell. We have another extension that is GPT in the ChatGPT that is called Skyler, and this one has the functionality to integrate with hardware as a microscope, and is now in a collaboration with a company, is like integrating and doing the control of the microscope. You can access the chatbot directly in the web page of the Bioimage Model Suite that acts as a help desk. Also, directly on the web page as a GPT in the ChatGPT environment. Now I will briefly describe the Bioimage Archive and how we are preparing artificial intelligence repositories to train models in the archive. So the Bioimage Archive is a cornerstone resources for the biomining community. It's a public repository that is stored in the EMBL and supports the storage and share of diverse biological image dataset. It's a centralized platform, and it empowers the researchers to access and contribute to the growing library of imaging data, fostering collaboration and innovation. The FAIR data principles are summarized in these slides and are transformative for both the scientific community and the individual researchers. For the community, FAIR data enhances transparency, boosts collaboration, and accelerates scientific progress. And for the researchers, increase visibility, secure datasets, and simplify the workflows, making a very valuable component of modern science. And remember, in the near future, FAIR data won't be a best practice, it will be a requirement. The repository perspective ensures that data is truly fair. For example, to be findable, data sets are registered with persistent identifiers and rich metadata. Accessibility means standardized protocols and secure access. Interoperability relies on formats and controlled vocabularies, while reusability requires clear licensing and comprehensive documentation. These principles ensure that data not only exists, but strives in global collaborative ecosystem. Breaking it further, repositories like the Bioimage Archive embody the FAIR principle by enabling rich metadata, registration, persistent identifiers, and machine readability, and they provide access to communication protocols, so support secure authentication, and promote the use of standard formats. And this is transforming how we share and utilize biomining data. The Bioimage Archive is free, it's publicly available, and support the biomining community by hosting these biological image data sets. It accesses data from any imaging modality, providing it's associated with a peer-reviewed publication or has value below a single experiment. It ensures that the data is fair, and it welcomes a wide range of data. It strictly excludes patient-identifiable medical data, maintaining compliance with ethical standards. And the Bioimage Archive simplifies data sharing and promotes transparency, collaboration, and innovation. The Bioimage Archive also houses a rich collection of AI-ready datasets that are tailored for machine learning applications in bioimaging. For example, these fluorescence images and annotations of high endothelial venules in tumor draining lymph nodes illustrate how these datasets are not only diverse, but also pivotal for cutting-edge research. By hosting such resources, the archive welcomes key enablers for AI-driven discovery, allowing researchers to develop, train, and validate models with real-world data. And to maximize the impact of this artificial intelligence-ready dataset, we must address the clinical data of image annotations. These annotations are vital for reproducibility, but they are hard to reproduce. And sharing and reuse these annotations are very valuable for advancing artificial intelligence methods. Also, these annotations are making fair to ensure that can be widely utilized and help to drive innovation. And collaborative efforts such as workflows and more workshops and shared repositories are essential to make this vision a reality. I'm gonna finish talking about this opportunity that are the open calls of the bioimage analysis support. That is driven by the AI for Life consortium. The open call is the chance to receive support in applying deep learning to the research. The deadline to apply is December 10. And whether you are struggling with data analysis, storage strategies, or custom workflow, the program is here to help. And applications are quick and are straightforward. Here is the timeline. They will be an application phase, then they are reviewing, consultation, announcement, and then the projects are running from beginning February to this summer. What do we offer in the open call? We use help in improving the image analysis workflow, improving the image analysis data or the data storage strategy, training, data creation, consultancy on available tools and solutions. And if we don't have enough resources to help you with the problem, then we cater the problem to other experts that have the resources to help you. Looking ahead, the future, as we envision the future of bioimaging, is as a collaborative effort. At the center of the vision is the integration of diverse resources like model repositories, like the bioimage.io, advanced analysis tool, accessible data repositories, and growing knowledge base. Add to this is the power of the artificial intelligence-driven chatbots and agents. And in this way, we aim to redefine how researchers interact with the technology. The Bioengineering is the infrastructure that acts as a connective hub, streamlining the workflows and enable the researchers to combine various resources effectively. And imagine the opportunity when a smart microscopy simply integrate with this artificial intelligence guided support for real-time feedback and decision making. Together, these tools and platforms lay the foundation for a vibrant ecosystem that fosters innovation and accelerates scientific discovery. And that will not be possible without all the team behind AI for Life and all the collaborators around the bioimage model zoo and the community partners. And we acknowledge the support from the European Union that has provided the foundation for this work. And with this, I will finish the presentation and I open to your questions.
Many thanks, Arrate, for this exciting talk. I particularly like the practical examples with the AI models. So yes, we have limited time before our coffee break. So let's go directly to one of our attendees. Veronica, you have raised your hand. Maybe you would like to ask your question directly to our speaker. Veronica, can you hear us? Okay, so let's go then to the Q&A box. I think, Arrate, you have you can see the Q&A box at the bottom of your screen where we have some questions from our attendees. I will read the first one. How many pictures will applications like BioPy need to train the model?
That will depend on the application, and that will depend also if there's a pre-trained model that is in the bioimage model zoo that is solving a very similar problem, or if there's a foundational model or a generalized model that is able to solve part of the task.
Great, thank you. The second question from Frank. With all these new tools appearing, is it correct to consider that the application of the FAIR principles is increasing and accelerating? How widespread would you say it is in the current situation?
Yeah, it's like the first principle, they are getting stabilized, and the adoption is accelerating. What I know more is like the community partners of the bioimage model zoo, that many of the open source image analysis tools and applications that are widely used are community partners of the bioimage model zoo, and there's also interest from companies. And that means that they are able at least to consume the models in the zoo, so they are adopting the standards and they are facilitating the reuse of the pre-trained models.
Thank you, Arrate. So the last question for you, how is the confidentiality of pictures uploaded in the applications or chat box? Will they be saved to train more models or deleted after use?
The images, they are not send it or share with us. The code of the chatbot that the chatbot generates is running directly in your web browser. So the images they never sent or transmitted to the cloud or to another server.
Great, thanks, Arrate. At this point, I would like to thank you very much for this exciting talk. I think we are going to have more discussions on this in the near future. Thank you very much for being here with us today. Thank you.
Thank you. Bye.